welcome back. In today's lecture, we are going to look at drug metabolism. So, this aspect of medicinal chemistry is very important, and because we will very soon see that even after we discover a drug and develop it, there are a number of things that can actually go wrong in during metabolism that can make a drug ineffective. It's also possible that a drug which is very effective in certain animal models may completely fail in humans, because the metabolism is very different in animals versus humans. So, therefore, understanding drug metabolism is a very important component of modern drug discovery. And if we have a good understanding of it, and if we can build good models, then it is quite possible that we may be able to avoid certain problems that commonly people face during advanced stages of drug discovery. Okay. So, just to sort of recap, we have looked at what pharmacodynamics is. Pharmacodynamics is nothing but the study of how drugs interact with a molecular target. Okay. So, for example, we have looked at on the cell surface there is a receptor. Okay. So, and to this receptor your drug is going to come and interact. Okay. So, how this drug interacts with the receptor to produce the desired signal or to inhibit the signal is something that pharmacodynamics deals with. Okay. While pharmacokinetics is another field on its own, is the study of how a drug reaches its target in the body. Okay. So, as you can very well imagine that just taking a drug orally doesn't ensure that it gets into where we want it to go and where we want it to hit. So, pharmacokinetics is a detailed study of how this happens, okay, and whether the drug reaches the target in the body and how much of the drug reaches in the body and so on, right. So, the main issues in pharmacokinetics that we are going to deal with, there are 4 of them. The first one is absorption, the second one is distribution, the third one is metabolism, and the fourth one is excretion. Okay. So, this is also called as ADME. Okay. So, ADME is a very important component of trying to understand how a drug or a drug like molecule functions at advanced stages of drug discovery. Okay. So, orally taken drugs, for example, we have looked at in the previous lecture that orally taken drugs have to be extremely stable, right, to survive the acidic conditions in the stomach, and also quite stable to survive the digestive and metabolic enzymes. So, we have already looked at the various conditions that the orally active or orally taken drugs are going to be encountering. Okay. They should be sufficiently polar so that they can dissolve in the gastrointestinal tract and get into the blood supply, but they should also be sufficiently have enough lipids or sufficiently fatty to be able to pass through membranes. So, both these criteria we have looked at in the previous lecture, right. So, at the end of it, it is for the drug to be orally active, it requires a number of parameters that need to be successful for it to be efficiently absorbed. Okay. So, we also looked at that most orally taken drugs follow the rule of 5, which was coined by Lipinski, and highly polar drugs can be orally active if they are small enough to pass through the cells of the gut wall, which are recognized by carrier proteins. We have already seen the example of how this carrier protein can bind to the drug and take it across the gut wall, or it can also go through by penocytosis. We have already looked at the mechanism of penocytosis, wherein there is an invagination that occurs, there is an encapsulation, and then it is passed through the gut wall. So, these are the mechanisms by which highly polar drugs can be absorbed. And we also saw that distribution of the drug is quite rapid, because rate of blood supply is very high. So, within a few minutes after entering the blood, the drug is going to distribute itself across the body. But the distribution is not even, because there are certain areas where the drug would preferentially distribute to over other areas. Okay. And another important component of it is whether the drug is bound to plasma proteins. So, we have already seen that plasma proteins can bind to a drug, and this binding interaction can be very strong sometimes. And what happens is that a lot of drug is actually trapped in the plasma protein. So, we have also looked at a case where when you take another drug, this drug that is bound to this plasma protein actually dissociates. And so, one needs to keep this in mind while thinking about distribution. Then some drugs we have already looked at have to enter the cell in order to reach the target. So, the examples that we looked at are enzyme inhibitors. So, if we design a drug as an enzyme inhibitor, then the drug has to enter the cell, meet the enzyme of interest, and then inhibit its activity, right. So, for which this drug has to enter the target cell. So, therefore, we have to keep this in mind when we are looking at how we are going to design the drug and whether it is going to reach the target and so on, right. So, in order for the drug to cross the barrier, the cell membrane, the drug has to have some level of lipophilicity, and that is very crucial for it to enter the cell, right. Now, a certain percentage of the drug may also be absorbed into the fatty tissue, that we looked at certain examples of that. When you have very lipophilic drugs, it is possible that they are going to dissolve in the fatty tissue. And we also looked at how the blood brain barrier forms a very very interesting situation, where not all drugs are going to enter the or are going to be able to cross the blood brain barrier. So, therefore, if we are designing a drug that is going to hit the brain, then we need to keep in mind that this molecule has to get across the blood brain barrier, or on the contrary, if we want a drug not to enter the brain, then we need to be able to figure out the right structural parameters wherein it does not cross the blood brain barrier. Okay. So, we have already seen that polar drugs are unable to cross the barrier unless they make use of carrier proteins or taken across by pinocytosis. And there is another interesting barrier which is called the placental barrier, which is applicable to expecting mothers. And so, this placental barrier sort of protects the fetus from many harmful chemicals, but the drugs can cross this placental barrier, and sometimes what happens is that this drug is remains even after birth, and it can prove toxic to newborn babies. Okay. So, with this background, let us now look at drug metabolism. So, most drugs undergo some form of metabolism inside the body, okay, and these resulting structures are known as metabolites, right. So, these metabolites sometimes lose the activity of the original drug. So, if the original drug, for example, was designed to interact with a receptor surface, let us say there is a carboxylate on this surface, and if the original drug was designed to have NH3 plus. Now, if this NH3 plus is actually metabolized to, let us say it gets oxidized to NO2, then that interaction is completely lost. Okay. But it is also possible that some of these metabolites will retain some level of activity. So, if the functional group change is not that drastic, then it is possible that some of the activity can be retained. In some exceptional cases, the metabolite may even be more active than the parent drug. Okay. Now, the reason why this metabolism is very important, one of the reasons is that a lot of side effects are actually associated with the metabolites and not actually with the drug. So, when the drug is tested through rigorous testing mechanisms, it is usually on a model system, and in the model system the actual drug may not be very toxic, but or even in an animal system it may not be very toxic, because the metabolism in animals is quite different sometimes from the metabolism in humans. And so, therefore, once the drug is now administered to humans, it is possible that certain side effects would result which are going to be deleterious. So, understanding drug metabolism will help with designing drugs that do not form unacceptable metabolites. So, it is also possible to take advantage of drug metabolism to activate the drug in the body. So, this strategy is known as the prodrug strategy. We look at this in detail towards a later part of the course. So, metabolism is divided into two parts. It is called phase 1 metabolism and phase 2 metabolism. So, we look at both of these in some detail in this lecture. So, as we can see, the body treats drugs as foreign substances, because invariably all drugs are going to be new to the body, and so it is going to treat it as a foreign substance. Now, in order to protect ourselves, our bodies have developed mechanisms to get rid of these foreign substances. So, these are considered as chemical invaders, and so the metabolism takes care of these chemical invaders. Now, if the drug is very polar, okay, so it is, let us say it is water soluble, then it is quickly excreted by the kidney. So, that is one of the things that happens. So, one of the mechanisms by which the drug is excreted is through the kidney if it is highly polar. But if it is not, then it is not easily excreted through the kidney, and it is here that drug metabolism occurs, where the fundamental reactions that happen invariably increase the polarity of the drug. Okay. So, the compound that is present inside our body is made into more polar molecules, so that they can be easily excreted. So, one of the largest class of enzymes that do this are a very non-specific enzyme, which is called as cytochrome P450, and they are present in the liver. Okay. So, one of the ways in which they act is to add a polar functional group. We shall look at it very shortly on a wide variety of drugs. Okay. So, then as we just saw that once you make the molecule more polar, it is then made water soluble and it is excreted through the kidney. So, now let us look at the reaction that cytochrome P450 catalyzes. So, you have a drug, let us say with a hydrogen molecule, and in the presence of oxygen, NADPH, which is a cofactor which we looked at earlier, and you need a source of proton. So, P450 acts on this and converts this to drug OH, and the byproducts are NADP and water. Okay. So, in the process, the drug has now been converted from, let us say, an alkane to a alcohol. So, this improves the polarity substantially, or increases the polarity substantially, and helps our system to get rid of it through the kidneys. So, an alternative set of enzyme reactions can also reveal masked polar groups. Okay. So, let us say there is a molecule which has a ester in it, right. So, now if you do a demethylation of the ester, then the ester is going to get converted to a carboxylic acid. So, the reaction that we are looking at is RCOOMe getting converted to RCOOH. So, this kind of a transformation is going to make the molecule more water soluble. Okay. And again, once you make it more polar, then it is excreted more efficiently. So, these reactions are classed into one phase of reactions, and they are called phase 1 reactions, and they generally involve oxidation, reduction and hydrolysis. So, most of these reactions occur in the liver, but some processes such as ester hydrolysis or amide hydrolysis can also occur in the gut wall or in the blood plasma and other tissues. Now, some of the structures in drugs are most prone to oxidation, and these structures are N-methyl groups. Okay. So, basically you have NCH3, okay. Aromatic rings, let us say you have a benzene ring, okay, and terminal positions of alkyl chains. So, let us say you have a long alkyl chain and you have a CH3 in the end, then this position is more susceptible to oxidation compared to the internal position. So, these are three examples of three functional groups that are pretty much more susceptible to oxidation. Other groups such as nitro group, which is basically N double bond O minus, nitro group, the azo group is N double bond N, right, and carbonyls, which is C double bond O, these are more prone to reduction. Okay. So, when a nitro group undergoes reduction, it is going to form R NH OH, which in turn is going to get converted to R NH2, and this can break down eventually by reduction to form R NH2, and a ketone can get converted to R an alcohol. Okay. So, these are some of the reactions that can occur, and of course, amides and esters, as we discussed earlier, are susceptible to hydrolysis by peptidases and esterases. So, we have looked at this these cases previously in detail, so I am not going to repeat this. For many drugs, it is also possible that two or more metabolic reactions might occur. So, you can have ester hydrolysis followed by oxidation of a different functional group, or you can have amide bond cleavage followed by reduction, and so on and so forth. Okay. And of course, there are also drugs which may not be metabolized at all. So, you have certain times functional groups which do not really react with some of the enzymes, and so they may not get metabolized at all. So, knowledge of the metabolic reactions that are possible for different functional groups allows us as medicinal chemists to be able to predict the likely metabolic products. So, for example, if I have a nitro group in my drug, then I can somewhat predict that it is going to undergo reduction, or if I have a ketone, then I can also suggest that it might get reduced to the alcohol, right. So, therefore, it is very important that we recognize this and understand how these processes occur. But this predictive value is also not that great, and it is possible that some of the metabolism that we may predict may not occur. So, only way to find this out is actually to do drug metabolism studies, which will establish whether these metabolites are really formed or not. Okay. Drug metabolism is also very important when you are looking at chiral drugs. Okay. We have spent quite a bit of time with examples on what chiral drugs are and how they can differentially interact with receptors. So, for example, a chiral molecule may interact with the receptor site in a particular way, and the opposite enantiomer may interact with it in a very different way, and so therefore, one needs to be able to understand how this chiral drug interacts. It is also possible that this chiral drug, this molecule with a chiral center, is actually used as a racemate, right. So, now if you use a molecule as a racemate, then we need to be able to individually test each of the enantiomers, and it is also possible that a chiral drug may racemize in the body. So, the presence of a chiral center creates certain issues that we need to deal with in detail in drug metabolism. So, the enzymes involved in catalyzing metabolic reactions will also distinguish between these two enantiomers, right. So, it is possible that one enantiomer interacts in a way that is very different from the other enantiomer, and not just that, it may give you completely different metabolites as well. Okay. So, when we are dealing with chiral centers, one needs to be sensitive to this situation and work accordingly. Okay. So, what we need to do is we would take both enantiomers of the chiral drug, and they have to be tested separately and to see what metabolites are formed. Okay. Now, in practice, it is usually preferable to have a single enantiomer, right. So, that is the role of asymmetric synthesis, or even better is to design a drug that does not have an asymmetric center, because if you do have an asymmetric center, then you are going to have these kinds of issues, and so you can develop compounds which do not have an asymmetric center. That means, you will make a compound which does not have a chiral center. Phase 2 reactions mainly occur in the liver, where the drug is actually conjugated with the polar group. So, the difference between a phase 1 and a phase 2 process is that the phase 2 you will have something coming and adding to the drug and making the entire molecule more polar. Okay. So, this results in increased polarity or water solubility. Okay. And again the same concepts apply as in phase 1, and you would increase the excretion rate in urine or in bile. Right. So, as you might have figured out, both phase 1 and phase 2 have very similar goals, that is to convert the less polar molecule to a more polar molecule. Okay. Now, phase 1 and phase 2 reactions can be quite species specific, that is something that undergoes metabolism in a particular way in a mouse model may be different from a primate model, and in the primate model may actually be different from humans. So, experimental animal models are important, but sometimes they are quite different from what actually happens in humans. Right. And this has also led to certain opportunities, because sometimes when a particular drug does not work in an animal model, we would still take it up to the next level, because it is possible that the drug may actually work in humans. Right. So, metabolic enzymes in different organisms can actually distinguish between identical functional groups or alkyl groups located at different parts of the molecule. So, this is the concept of regioselectivity. So, you can have a metabolic enzyme which is regioselective to a particular area of the molecule, while the other metabolic enzyme can be regioselective in the other region. Right. But of course, you can also have the stereoselectivity, where you have different stereoisomers inside the in the same molecule, or a single stereoisomer where one metabolic enzyme recognizes one stereocenter while the other one does the other way in the other case.